Biological aging in oocytes creates a divergence between the cell's chronological age and its metabolic capacity. We track the finite inventory of oocytes as a numbers game, but the actual limiting factor is the dynamic degradation of cellular health. The baseline numerical depletion follows a predictable timeline. A female is born with approximately 1 to 2 million oocytes. By puberty, that reserve drops to around 300,000. Clinical outcomes in reproductive medicine highlight a focus beyond the sheer quantity of eggs remaining. The critical metric is the functional competence of these cells as they age. This chart maps these two competing timelines. While the descending line shows the depletion of the ovarian reserve, the ascending line tracks single-cell sequencing data. As the numerical count drops, the rate of aneuploidy surges from 14.5% to 38.3%. Aneuploidy occurs when a dividing cell fails to distribute its 23 chromosomes evenly, serving as the primary driver behind IVF failure. We track this age-related decline as a sequence of metabolic and epigenetic failures that can be identified and measured at the single-cell level. The oocyte is the largest cell in the human body. Unlike a sperm cell delivering a genetic payload, the oocyte carries the entire cytoplasmic machinery required to support the first critical days of early embryo development. For an egg to be viable, it relies on two entirely distinct biological systems, executing their roles perfectly in tandem. Nuclear maturity describes the precise alignment and segregation of the 23 chromosomes required for genomic integrity. Cytoplasmic competence measures the health of the organelles and the density of the energy-producing mitochondrial network surrounding the nucleus. The nuclear machinery is entirely energy-dependent. If the cytoplasmic power supply fails, the physical force to separate chromosomes is lost, resulting in chromosomal error. The physical separation of chromosomes during cell division, known as meiosis, places an extreme metabolic demand on the oocyte. Pulling these dense structures apart requires massive volumes of cellular energy, or ATP. At a microscopic scale, chromosome segregation is a violent, highly kinetic event. The cell must assemble a physical apparatus, lock onto the chromosomes, and forcefully drag them to opposite poles of the cell. With age, the mitochondria responsible for generating this power begin to fail. Their physical morphology changes, mitochondrial DNA copy numbers drop, and overall ATP production plummets. This failing energy grid creates a highly toxic byproduct, reactive oxygen species, commonly known as free radicals. Young oocytes neutralize these molecules efficiently, but aging reduces the cell's ROS scavenging enzymes. Without this defense, free radicals accumulate rapidly, actively damaging the delicate DNA and lipid membranes of the cell. The collapse of the antioxidant defense system means the oocyte's own energy production process turns into a toxic cascade that degrades the cell from the inside out. The ATP deficit directly sabotages the meiotic spindle, the physical, mechanical sorting machine responsible for pulling the chromosomes apart. Without sufficient energy to power the spindle machinery, the assembly process becomes unstable. Chromosomes are misaligned, and when the spindle pulls, it missegregates them. This structural, mechanical disruption is the physical root cause of the 38.3% aneuploidy rate we see in aging cells. The cell simply runs out of the energy needed to sort its DNA correctly. The damage compounds. The accumulated oxidative stress accelerates the shortening of telomeres, the protective caps on the ends of chromosomes. This attrition strips the chromosomes of their physical protection, driving further genomic instability. Metabolic failure dictates structural genetic collapse. The drop in ATP and the rise in oxidative stress directly cause the machinery of the cell to break down. This degradation is not contained to the nucleus or the mitochondria. It spreads to the cell's external boundaries and its signaling pathways. The zona pellucida, the outer protein shell of the egg, begins to physically harden, obstructing sperm penetration. Simultaneously, the endoplasmic reticulum loses its capacity to store calcium, blinding the egg's ability to respond to fertilization signals. At the genetic level, aging alters the cell's DNA methylation patterns. This epigenetic dysregulation means environmental factors begin turning off specific genes that are mandatory for healthy embryo development. Recent single-cell resolution studies have identified the targeted downregulation of two highly specific genes, CALB1 and RPL23. The loss of these specific gene expressions initiates a severe feedback loop, driving further mitochondrial dysfunction and guaranteeing higher levels of reactive oxygen species. While an oocyte is as old as the person carrying it, the cell undergoes a final 90-day maturation phase before ovulation. During this 3-month window, the cell's cytoplasmic competence is highly responsive to its microenvironment. Clinical interventions during this maturation phase optimize the metabolic environment surrounding the oocyte, providing the fuel necessary for meiosis. Coenzyme Q10, specifically in its ubiquinol form, acts as a primary electron transporter in the mitochondrial respiratory chain. Clinical data shows that consistent supplementation before IVF retrieval reduces abnormal mitochondrial distribution in aged oocytes by 35%, directly correlating with improved embryo quality. Similarly, a 2025 systematic review from University College London found that nicotinamide mononucleotide, or NMN, actively restores cellular energy metabolism by boosting NAD+. This powers the sirtuin proteins responsible for protecting mitochondrial DNA. These interventions are targeted, biochemical strategies designed to stabilize the oocyte's energy grid in the precise window before ovulation occurs. This metabolic support extends to an antioxidant arsenal. High concentrations of vitamins C, E, D3, and melatonin act as direct scavengers in the follicular fluid, neutralizing reactive oxygen species before they can damage the cell membrane. Hormonal precursors like DHEA further support the intrafollicular environment, improving ovarian response and establishing a baseline for healthier oocyte maturation. The 2025 data moves beyond supplementation and into the territory of active cellular restoration. The focus of emerging science is ensuring the successful mechanical execution of chromosome segregation in aging cells that would otherwise fail. Research on plant compounds like echinococide has demonstrated the ability to physically restore spindle morphology and preserve mitochondrial membrane potential via targeted cellular pathways. Furthermore, the targeted overexpression of the CALB1 and RPL23 genes has successfully rescued age-related defective phenotypes in advanced models. Oocyte degradation is a specific sequence of metabolic and structural failures. By defining these mechanisms, reproductive science treats cellular aging as a modifiable condition with concrete